Another type of microbe that's included in the unit we're studying are viruses. Now, viruses are a little different from some of the other microbes we've talked about because viruses, by most biologists, are not really considered to be alive. They don't meet some of the basic conditions of living things. In other words, they don't have cells, they don't have their own metabolism, so they really don't comprise another kingdom like animals, plants, fungi would. Anyway, we'll just call them infectious agents. And in order to replicate, they need to use the metabolism, the machinery of some other living thing. So they need to replicate inside a living organism. And for some reason or another, some viruses do very well in certain living organisms, but not other living organisms. And that's why they often have names like bird flu or swine flu, because those were the living things that they seem to replicate best in. Viruses are quite simple. In general, they're actually made of three different things, and those three different things are nucleic acids. Now, nucleic acids could be DNA or RNA, and all RNA is single-stranded, but the DNA inside a virus could be single-stranded or double-stranded, which is very interesting. And that's why sometimes with viruses, Chargaff's rule doesn't really hold because they might be single-stranded. So we have some genetic material. There it is inside there. And then it's enclosed in a protein coat. So we've got some sort of protein coat. Sometimes it's called a capsule or a capsid. And then outside the protein coat is kind of a lipid layer. So you can see the lipids out here, and that gives it a certain stickiness and helps it to attach to things. And as we'll see, that most viruses are very specific for the things that they will attach to. So once they get inside, then we have to replicate. So they have to use the metabolism of something else. So here's basically how it goes. We have three steps. First of all, they have to attach, and they don't attach to a lot of things. They're very specific. For example, the HIV virus will only attach to T cells, which is part of our immune system. We'll learn about that later on. And once they attach, then they penetrate, which means that the genetic material goes inside the organism, the cells that they're trying to infect. And then they go ahead and they use the replication machinery of that cell to multiply, so replication. And there are several different ways they could do it. So they use that to make lots of copies, right? And then those copies are translated, again using the machinery of the cell, into proteins. So protein synthesis, and then they're assembled just like the virus was before. And then sometimes they can sit around for a long time and not do anything. This is called the lysogenic cycle. Or they can go into the lytic cycle. For some reason, something sets them off, and they destroy the cell, and they release themselves, millions of copies, and then they go on to infect somebody else. So DNA versus RNA. Okay, DNA viruses. How would they replicate? Well, DNA viruses would go straight into the nucleus, and they'd pretend that they were just DNA, just like the host DNA, and they would be transcribed into RNA, just like the chromosomes would be, and then they're made into viruses instead of the proteins that the cell wants. What if you're an RNA virus, though? How would you go ahead and do it then? Well, what you would do is you would just stay out in the cytoplasm and pretend that you're a piece of mRNA, messenger RNA, from the host nucleus, and just wait for the ribosomes to come along and translate you into a virus. And then we have a very interesting class. We have some RNA viruses who can do a different sort of a trick. When they get injected in, they also inject an enzyme called reverse transcriptase. And what reverse transcriptase does is it takes RNA and it makes it into DNA. And then the DNA will often insert itself into the chromosomes of the host. And it'll just go along for the ride. This is what HIV does, and this is why it's so difficult to get rid of it, because it actually becomes part of your DNA. Okay, well, viruses are really responsible for a lot of terrible epidemics. And an epidemic is something that kills a lot of people locally, but sometimes they actually go global and then we actually call them pandemics. Now, a lot of diseases you're already familiar with, like the common cold, not too bad. I wouldn't call that a pandemic, but some things are. Here's a couple of them you should probably know about: AIDS, HIV. That is caused by a retrovirus, an RNA virus, and it has infected currently about 40 million people, although we'll talk about it later. There's been a lot of drugs lately to kind of keep it under control. Hey, one that's been in the news lately has been Ebola, which has hit into West Africa in 2014. Nasty, nasty disease. But probably the big killer of all time was one you wouldn't really think about: the good old flu, influenza. In 1918, we had a terrible strain of flu called the Spanish flu, which hit the United States pretty hard too, and Europe, and killed probably worldwide 50 million people. Now, why do we keep having strains of flu? So the Spanish flu, and then you had SARS, bird flu, and Hong Kong flu, and so forth, because most viruses, since they're so simple, mutate very, very quickly, and they change. And so you can keep coming up with vaccines for what you think is the most likely variety of the virus, but pretty much by next year it's changed and a new one just comes along. And you got to go ahead and get inoculated all over again.